Well, my name is Grace Quartzen. I'm a member of the Explorers Club, and I'm also the leader of the next generation of exploration within the Explorers Club. What is NGEN, or that abbreviated? Well, NGEN is roughly the members under 35 to 40 within the club, but that's really who we're highlighting. We really welcome everyone to be a friend of NGEN who is a member or not a member. But the group NGEN exists to highlight what people are doing. And that's exactly what we're going to do today, is celebrate some of the members within the Explorers Club who are just starting off in their careers or are in that age group and working on things in that way. What else NGEN does in the club is we have vibrant online community for those younger members all over the globe. We do speaker series like this at a variety of Explorers Club events throughout the year, and then other meetups, casual social things here and there at chapters all over the world. Welcome to an amazing event today. This is the NGEN Symposium. It's been going on since 2019 and has grown and grown every year. What started as just a few people in the audience and a few NGEN members has become what we're seeing today here at the Asia Society, and we're so excited to have you. We received an incredible group of applicants this year, and I am excited for you to hear from every single one of them. And so to kick things, let me first tell you how it's going to run. So each speaker is going to come up. They're going to get roughly 8 minutes to tell you about their work and what they've been up to, and then we'll have 2 to 3 minutes of question and answering before moving on to the next speaker. All of this is going to be moderated by the amazing Alex Borowitz, who's an Antarctic ecologist and fellow member of the Explorers Club and one of our amazing event planners as well. Before we begin, I want to say a huge thank you to the Explorers Club, as well as Alex, Lizzie, and Bree, who we've all spent many months organizing this and putting it together, and I'm so excited for you to hear from our amazing speakers. So without further ado, I'm going to hand things over to Alex, and I hope you enjoy the NGEN Symposium. Thank you.
Thanks, Grace. I love the NGEN Symposium so much, and I'm so excited you are all here to join us and celebrate these awesome folks. Our first speaker is Monique Sosnowski, and I met her, I think, in 2019 at a conference at the American Museum of Natural History, where she gave, I think, the most accessible and entertaining talk on illegal fisheries that I think I've ever seen or am likely to see in the future. Monique is, her area of research is wildlife crime, which is to say things like trafficking and poaching and not crimes that animals commit. She has worked with universities, governments, NGOs, all sorts of folks doing training, providing innovative, data-driven solutions to wildlife crime. She is a prolific scholar and holds a PhD from John Jay College of Criminal Justice. So I'm very excited to introduce you to our first speaker, Monique Sosnowski.
Thank you so much. Hi, everyone. As Alex said, my name is Monique Sosnowski, and I explore the hidden networks behind wildlife crime. So today, I'm hoping to show you how next-generation explorers are combining conservation science, criminology, forensics, and big data alongside next-gen technologies to combat trafficking, protect our ecosystems, and redefine what it means to explore. Exploration isn't just about going where nobody's been. It's about uncovering what others have overlooked and looking at things from new perspectives. Wildlife trafficking isn't just a conservation issue. It's a crime. And solving it means crossing boundaries between disciplines, between countries, and between what we think science is and what it can become. My approach blends conservation with criminological theory, forensics, and big data to explore how wildlife crime and trafficking happens. We're not just studying the animals, we're tracing networks, we're analyzing behaviors, retraining how people think, and thereby helping law enforcement stay one step ahead of organized environmental crimes. What does next-gen exploration look like for me? It's working with law enforcement and conservation officers from South America through Africa, from the Andes to the Okavango Delta. For years, there has really been a divide between conservationists who have been traditionally trained to approach wildlife conservation issues through ecologically or more biologically informed lenses, while on the other hand, law enforcement has been trained strictly through traditional law enforcement and paramilitary practices. My goal is to change this, to promote a new, interdisciplinary, data and evidence-driven approach to problem solving. So I train law enforcement and conservation professionals to see wildlife crime as this type of an interdisciplinary issue. Whether it's trafficking parrots in Peru or snaring across South Africa, my goal is to reframe the crime as well as to rethink our responses. So these are just several images of me this past December training parrot trafficking crime prevention workshop in Brazil. My approach is rooted in a criminologically rooted foundation. I apply criminological theories as well as techniques to identify crime opportunities, to explore how traffickers are really weighing risks and benefits, as well as to look at things like seller behavior, seller profiles, or online encrypted communications. Along this, I map trafficking networks and much more. What my goal is, and what's really important, is that we're really no longer guessing. We're using evidence-based approaches to investigate with a purpose. One of the techniques I use in the field to do this is called crime scripting. It's the development of what can be best compared to as a movie script that is rooted in understanding crime patterns. So they're really fun to put together. But together with law enforcement and conservation teams, we map out step by step from capture all the way through consumers and ask questions like who's profiting from these crimes, what tools and technologies are used to capture animals, to prepare them, to sell products, where is our law enforcement weak, where are the gaps? And in one case, for example, activities like these with large teams have helped us understand that jaguar trafficking was involved in convergence with drug smuggling routes. So this is exploration through interdisciplinary systems thinking. I had mentioned data. Data really helps us see the bigger picture. It helps us understand these systems and the extent of wildlife trafficking as a global crime. By gathering and analyzing data from nuances in human behavioral patterns through the routes that wildlife products are taking, we can create maps just like these to better understand what our next steps in crime prevention can be. But law enforcement faces rapidly changing tools and technologies today. In response, our crime-fighting technologies have had to also advance. Today, we're not only deploying drones with infrared detection to monitor our parks at night, or using satellite surveillance to catch illegal logging, for example, but we're using advanced geospatial technologies and softwares to actually predict poaching patterns based on our data. We're examining blockchain to track supply chains and explore how poachers and traffickers are using cryptocurrencies. And we're doing things like working with computer scientists to develop AI and machine learning algorithms that help us scan the web to flag suspicious sales. And this approach isn't just meant to build cases, it builds capacity. One officer this past December told me that these methods allow everyone to see that they have many problems in common when it comes to parrot trafficking, and that the modus operandi, or how these crimes are being committed, is very similar across countries. This is exploration with a multiplier effect. It's not just investigating wildlife crime, but it's transforming how the world thinks about it and how the world fights it. And the world needs to fight this now more than ever. Today, organized criminal syndicates are increasingly known to be involved in wildlife crime. From the Pablo Escobar of animal trafficking operating out of Laos, from organized criminal syndicates operating out of a casino in the Golden Triangle, to a slew of Mexican organized crime groups extorting small fishing communities across Latin America, these investigations get larger, they get more complex, and they get way more hands-on. Knowing this, we need all hands on deck. We must break out of our silos, like many of us here in the NGEN community are doing, to collaborate. From conservationists to criminologists, forensic scientists, computer scientists, data analysts, and many others. But we also need banks that are being used to facilitate illegal transactions, or the social media platforms and websites where illicit ads are being posted for wildlife products, alongside the public to support this kind of work. After all, this is a global fight, and there is a role for everyone. Support your organizations that fight trafficking, report suspicious online wildlife behavior, and push for stronger policies whenever you can. Whether you're coming from science, law, technology, advocacy, or something different, you can be part of this solution. So to close, there's so much for us to fight for. We're not just fighting to solve cases or make arrests or have product seizures, but we're really fighting for hope, we're fighting for freedom and a healthy planet that supports all life and the survival of our species, many of whom teeter on the brink. So with that, thank you so much for your time and attention today, and I would be happy to field any questions.
Thank you so much, Monique. So here's how we're going to do questions. We have a few minutes here. We've got a couple of microphones, so raise your hand, we'll call on you, but before you ask a question, please wait for someone to arrive with a microphone so we can all hear you. Anyone starting first? All right.
Where do the confiscated items from these criminals, whatever you want to call them, end up? I think Smithsonian might have a place for those. I think there's other places in the States, are there not, where they confiscate these illegal trophies, so to speak, that come into the country.
Yeah, that's a fantastic question. There's a couple of different options for products that are being confiscated. Live animals are often worked out with zoo partners that might be in countries where those products or where those animals are being confiscated. But other products that are no longer living might be there's a U.S. Fish and Wildlife warehouse out west that has hundreds of thousands of seized products, but other products, like you mentioned, do end up in museums and are used for educational purposes and science, essentially, for the public. Ah, perfect.
Hi, could you share with us some of your big wins and then some areas that you feel are worthy of further work in the next year or two that you're focusing on?
Yeah. I think some of our biggest wins have been working to bring together law enforcement and conservation professionals in the field. That slide that I had near the beginning where we were able to bring together law enforcement and conservation professionals from essentially all countries across South America, put them all in one room and have them all talk and sit together and work through activities for multiple days and come away actually saying, wow, we have the same problems and we can share solutions and pool together our resources to fight this together instead of being alone is incredible. So I think in the large scheme of things, it's about improving our law enforcement and conservation team communications. It's about pushing our technologies forward and having the funding to actually do so, and really bringing these crimes to the attention of judges and courts and policy decision makers who often in a court where they're prosecuting homicides next to human trafficking and end up with wildlife crime cases aren't necessarily going to see fish smuggling as a huge problem when they're really not looking at, like I said, this bigger picture of this being a global, often organized crime issue. So I think that those would be the areas that would require the most kind of push forward and funding and support.
I think we've got time for one more.
How much is this sector actually worth? I mean, if you can compare it to other, is it $100 billion? What's the market share?
Yeah, it's of course extremely difficult to measure this. It is ranked the 4th largest transnational organized crime, so it's essentially up there with drug trafficking, human trafficking, and arms trafficking. They estimate that it's worth up to $200 billion a year, depending on if you're including illegal fishing and logging industries. But it's a massive, massive network of individuals around the world that are participating in the more organized networks. And again, they're heavily, heavily intertwined with arms trafficking, human trafficking, and drug trafficking as well, which is something that we're seeing more and more, given that this area also doesn't necessarily get as much attention or as many resources when it comes to detection and enforcement.
Awesome, thanks for those questions, and let's all thank Monique one more time.
Thank you so much.
I'll say it again, I love the NGEN Symposium so much, and part of that is because our young members do so many interesting things, but not just interesting things, but things in new ways. You know, exploring with new methods and you know, breaking down old barriers. And our next speaker is another one of these people. Zeff is a PhD candidate at the Global Change Research Group at UC Santa Cruz, and she is a marine community ecologist, but also an environmental poet. She works to understand how climate change changes the interactions you find between species in the ocean. And her work has really brought her around the Pacific, studying reefs and food webs, and doing science that's community-oriented and solutions-driven. So in short, science that's not just for scientists. When she's back on dry land, she also facilitates eco-poetics workshops and builds discussion about the role of researchers in society. So I'm very excited to welcome Zeff to the stage.
Yorana Tato. Hello, everyone. My name is Zeff, and I'm a marine ecologist, which means that I spend a lot of my life underwater, as I am here, surrounded by this group of striped Salima in the Galápagos Islands. And as an ecologist, I'm not just interested in individual species or isolated environments. I'm really interested in the web of interactions that actually connect them. And these relationships and these connections are what build resilience in the marine ecosystems I care so much about. And so my research is really about understanding those connections and what causes ecosystems to thrive or struggle when conditions change. I've worked in the Galápagos Islands for around 7 years, and that's normally where you'll find me, but today we are going to travel across the Pacific to another island that's very close to my heart. So when I first arrived in French Polynesia on the island of Moorea, like everyone else, I was amazed by the dynamic volcanic peaks above and the coral reefs that were extremely vibrant below. But as I continued my fieldwork, I got to discover something that a lot of visitors don't get to see, which is a network of freshwater springs that actually run underground and flow from the mountains to the reef. And we call these hidden waterways the veins of the island. Let's talk about how these freshwater springs work. Rain falls on the mountain peaks, filters through the porous rock, and emerges underwater, bubbling up on coral reefs and creating what's called submarine groundwater. And submarine groundwater is a completely natural phenomenon, but it can really easily carry contaminants from human activities such as agricultural fertilizers and pesticides, highlighting why it's really important to understand the biogeochemical balance that actually sustains healthy reef communities. So I study these systems by deploying submersible water samplers that are taking water samples throughout the day and the night and helping us understand the unique chemical signature of groundwater on the coral reef. And what my team found is that these freshwater inputs aren't just adding water to the ocean. The nutrients that are found in groundwater is actually boosting photosynthesis and changing the water chemistry and making it more acidic. And that's really affecting the corals and other organisms that need to build their own skeletons. Using respirometry, which is basically just a way to measure how corals breathe, I found that corals that are from low groundwater environments are consistently outperforming those from groundwater-rich environments, which tells me one thing that's really important, which is that corals are carrying their environmental history in their physiology, and these adaptations persist even when conditions change, suggesting that how different coral communities respond to climate change is going to largely depend on the environmental conditions that they've been used to or evolving with for generations. Perhaps most surprisingly, I found that groundwater creates these really unique fish communities where estuary species like these barred flagtails are swimming alongside and mingling with classic tropical species. So it was kind of like seeing Dory and Nemo hanging out with fish that you would expect to see in a river mouth. It was very strange. And a lot of local Pacific Islanders actually traditionally harvest these flagtails for food, which I think shows the real importance of making sure that the groundwater is clean, not just for the reef communities, but also for the people that depend on them. So I hope by this point I have convinced you that groundwater is a very powerful ecological force that structures coral reef systems across multiple scales, from water chemistry to community composition, all the way to ecosystem function.
Water in Polynesian, it's called vai. Vai means also to live. Water on land, it's like blood in your body. When the land and the sky, they meet together, Sky Father is crying and the rain falls on Mother Earth. She funnels it through a spring or through a river where it will meet the ocean. Sometimes it will also go underneath the sand and comes out in the lagoon. That pathway for us is groundwater, is vai, is life.
As you just heard from respected cultural scholar Hinano Murphy, these freshwater systems have been known and valued by Polynesian communities for centuries, and many sacred sites around the island are located near these freshwater springs. So recognizing the value of this knowledge, I co-founded the first of its kind cultural exchange program between the University of French Polynesia and the University of California, Berkeley, in Moorea, the research station, the first collaboration there that is a cultural exchange program that is empowering Tahitian students to learn their desired scientific skills and also enhancing our understanding of traditional perspectives on water systems. And beyond the science, I've also been able to learn songs, stories, food, dance, weaving, learning to prepare practical cultural elements like pasang cru or fafaru, which are traditional fish meals that represent the relationship to the marine environment. And this kind of integration didn't just enhance our science, I think it really completely transformed it. And I love this quote by Tamiri Lowe, one of our cultural exchange students, which I think exemplifies the outcomes of this program. I used to think coral were rocks. I didn't care when I would surf over them. Now I know they are alive, just like me. And this program's fostered a new generation of environmental stewards from both French Polynesia and the United States that don't see science and culture as separate domains, but rather complementary ways of understanding their island home. And this was an immense group effort, so before I leave you all today, I'd really like to thank the Mahe family, the University of French Polynesia, and UC Berkeley Gump Research Station, as well as Tamiri and Hayata, some of our students, the Revatea and Atithia centers, and of course the Silberger Lab, without whom this research would not have been possible.
It's like you growing a home where every wall talks to you. It's like every molecule of the water is speaking to you. If we don't rebuild this relationship of the water, the principle of life is not sacred anymore. We are all taking from Mother Nature. Let's all give back a little bit by putting our efforts together. If you take care of your water, the water will always be there for humanity. How can I say it's for all of us? We say in Tahitian, no tatu. It's for all of us.
Just as freshwater springs connect the mountains to the coral reef, I really want my research to demonstrate how connecting diverse knowledge systems can build stronger and more resilient approaches to environmental challenges. And by combining scientific rigor and cultural wisdom, we can start to understand not just how ecological communities are functioning, but really why they matter. And a lot of the relationships I've talked about today are facing mounting threats due to global change, but by bridging science and culture, I believe that we can protect the veins of Moorea and islands everywhere for generations to come. Marururoa, thank you so much for listening.
Wow, incredible. Do we have any questions for Maya?
Thanks for such an amazing talk. I was wondering if you have any plans for future work and where you're thinking that you're going to take your research program in the future, like building off of this kind of stuff.
Yeah, right now I'm working mostly in the Galápagos, but really trying to get back to French Polynesia as well. I think one of the main things for this specific research is trying to understand how much groundwater is good or bad, both for the reef and for the people, because as I said, those contaminants can just come so easily into the coral reef community where it's just a few feet away from where people are living. And so one of the things I'd really like to do in the future is be able to work with individual homes and also test water and make sure that they know where the groundwater seepage points are next to their houses and what they're using for food and how that's translating to their own health and their families, because we got really close with a lot of families near where we were working, and I'm interested in their kind of well-being long-term. More longer term in terms of my own research program, I'm just really interested in using network approaches to understand the configuration and structure of species interactions. So we're mapping out food webs in the Galápagos right now and trying to understand how temperature is impacting their structure and what that means for the stability of those systems. So that's the plan. Thanks.
I think we don't have time for another one, but please do find Maya afterwards and ask more questions. But let's give her another round of applause. Thanks so much. All right, we are going to stay underwater for a little while. Our next speaker, Hailey Shchepanik, is a member of the Canadian chapter, and she's a diving safety officer at the Vancouver Aquarium and also an undersea specialist with National Geographic Lindblad Expeditions. Her work has focused on the Canadian Maritimes and the Arctic, but she's done research across Canada and in Belize and in Saudi Arabia. She has a master's degree in marine science from King Abdullah University and has gotten awards from places like Wings and the Women's Divers Hall of Fame. So I'm very excited to bring Hailey Shchepanik up.
Hello, everyone. My name is Hailey Shchepanik. My background is marine science, and I work as a biologist, an expedition diver, and diving safety officer. This is my very first time attending ECAD, so it's really exciting to be here for the NGEN Symposium today. This project that I want to talk about today was one of my favorite that I've worked on thus far, and it was part of an opportunity that I had with the Our World Underwater Scholarship Society. And this organization has been dedicated for the past 50 years to providing really excellent opportunities for the next generation of ocean professionals. And today I thought I'd talk a little bit about how I ended up in the small town of Red Bay on the east coast of Canada, working underwater on some of the world's most well-preserved Basque whaling shipwrecks from the 16th century. So Red Bay is located on the southeast coast of Canada in Labrador, and it has a population of 150 people. However, we now know that from the early 1500s to mid-1600s, thousands of people traveled across the Atlantic to come here to hunt right and bowhead whales. However, the exact location of these whaling stations in Canada were largely unknown until the 1970s, and this is when archival researchers began to uncover archaeological sites and pinpointed the location of several really well-preserved and impressive shipwrecks. So what brought us here to the site was the wreck of the San Juan, and this was a 30-meter-long whaling vessel that sunk in 1565 with over 1,000 barrels of oil on board. And it wasn't until researchers found an insurance claim document from when she sank that they actually were able to pinpoint its location and start the investigations. So this is just a little bit of background. This happened, the original excavation happened in 1979, and it took Parks Canada staff 7 years to complete the excavation. So this is one of the largest underwater archaeological projects in Canadian history. And with all this information that they were able to gather, since the wreck was so well preserved, they were able to fill some really long-standing gaps in our knowledge of evolution of shipbuilding technology and Basque whaling history. It was even featured on the cover of National Geographic in 1985, and then resulted in the establishment of UNESCO World Heritage Site in 2013. So this is me here in 2022 with the Parks Canada Underwater Archaeology team, and we came to do their annual site assessment and maintenance of this UNESCO World Heritage Site. So for me, this represented my first opportunity to dive in waters ranging from -2 degrees Celsius to +2 degrees Celsius. And it was also the first time I was able to use a full face mask and underwater communications, a bailout tank, and all of these different new pieces of gear, even the neoprene dry suit and the lobster gloves we had to use underwater to carefully sew zip ties. Those were all new to me. But this project really quickly solidified my love of cold water diving. I just absolutely love being in these sorts of environments. And it was also a really fun challenge to try to keep up with the commercial divers on this project in really limited visibility and cold conditions. So underwater, I don't usually use a crowbar, but this was a project where we were doing a lot of heavy lifting, and our aim was to repair the protective tarp that now covers this site. Because even though during the original excavation, each and every of the 3,000 timbers were carefully excavated, brought up to the surface, and documented, they were all eventually put back, covered with a layer of mud and a giant tarp in hopes of recreating the homeostasis and the anaerobic environment that kept this shipwreck so well preserved for hundreds of years. So this project has strong links to our continued understanding in Canada of our natural and our cultural resources. You can see some timbers that weren't reburied in the top left corner, and the archaeologists were actually able to reassemble a chalupa and understand its importance as the sort of support vessels for these whaling expeditions, and really understand what the Basque whaling techniques would have looked like. And also, the detailed information gathered from this excavation has actually taken more than 30 years to kind of go through and analyze. And all this information has been put to many uses, including actually recreating a life-size replica of the San Juan, which they're going to sail from Spain to Canada this year. It's been a work in progress, so they're finally almost there. But they're using the traditional methods that had been missing from written records that this excavation was able to uncover. On the natural history side of things, there's almost an estimated 25,000 whales that were hunted and killed at this location, and there's an abundance of whale bones at this site, so researchers are using ancient DNA to try to understand species history, reproductive potential, and to inform recovery efforts of this now critically endangered species. So this project was just one of over a dozen that I worked on with our World Underwater Scholarship Society. And even though I played a really small part in each of these projects, I wanted to talk about it today because it's played a really big part in kind of shaping my career over the past several years. I actually was able to return to Red Bay every single year since this project, and knowing that it takes like 5 flights, 2 cars, and a ship to get there, I really didn't think that I would be able to go back. But now I get to go back each year as an expedition diver and a naturalist, and I get to give lectures and share my experience in this region. Most importantly for me, this opportunity made me realize that my eventual career goal was to become a diving safety officer. And I was a recent graduate at the time. I thought that this was like a distant possibility, but the support of the Scholarship Society, the National Park Service, and the Explorers Club has really leapfrogged me in my career, and now I get to say that I'm one of the youngest dive safety officers in Canada in a role I just started a couple months ago. I think something else I realized was kind of a reoccurring theme that a lot of us in marine science experience on a daily basis, the fact that the contents and the changes of our oceans has historically gone quite overlooked. And it's kind of interesting to note that this shipwreck was found in just 12 meters of water. So for 500 years, it went unnoticed, and it's a stone's throw away from the town. So kids would quite literally pick up stones, skip rocks, but these stones that they were using are actually tiles from that the Basque whalers brought as ballast in their ships and left in Red Bay. So again, time after time, the shipwreck was just standing there and we didn't know. So this is kind of the idea that kind of drives my continued desire to pursue a career as a scuba diving instructor, as a dive safety officer, as an expedition scientist, is I want to, hopefully be able to give scientists and the general public the tools that they need to care for and explore our oceans. Thank you so much for joining the symposium today as we highlight how just incredibly valuable it can be to make space for NGEN in research projects and expeditions. I know for many of us, these opportunities can have a snowball effect that really catapult us towards our career goals. So thank you very much.
Have some time for a few questions. It looks like we have them.
Hi. Was there any shipbuilding technology that was not known right now that was found in at the shipwreck?
Yeah, absolutely. So I don't know the ins and outs of how each timber assembles and what that meant for like the information that was gathered. But yeah, this ship in particular just was in such good condition that they were able to understand like more details about the evolution of shipbuilding. So all the Basque whaling, the lack of written records, this ship kind of is now an example of the techniques that they used. And that picture that I showed about the recreation that they're doing in Spain, it's quite interesting because they're using like traditional methods that, again, like the methods that have been pulled from this excavation in terms of like knowing that you should only cut the wood like at a certain time of the month when the moon is like at a certain point of the horizon, and like exactly how the keel and all these things kind of fit together. So yes, they definitely learned a lot in terms of the shipbuilding methods that the Basque were using based on this site.
Thanks so much. Another question?
Very good talk, thank you. Can you just share with us where these whalers came from? We're talking about the 1500s. Were they European, native Canadians? And
Yeah.
Another curious question, was any of the whale oil still intact in containers?
So these whalers, they came from what's like present-day Spain and France. So they traveled across the Atlantic every year and stayed for like a couple months and then would go back with their haul. I think it's amazing to know that like this ship sank with 1,000 barrels of intact whale oil, but during the excavation, there was no intact barrels remaining. So there's a couple pictures. There's like a 5-volume series on the excavation that Parks Canada gave to me. It's like in my personal library, but in there you have pictures of the like kind of flattened casks, and the entire ship just kind of like opened up like a book. But yeah, no like intact whale barrels were found, I believe.
How do the anaerobic conditions in Canada compare to the Baltic and the Vasa and things like that?
So I'm not exactly sure how the preservation of the many shipwrecks that were found in the Boss, but this shipwreck was covered in like a foot of mud when they found it. So that's what kind of created these anaerobic environments over time. Of course, the cold water did help, but it was primarily the mud that prevented different biological, mechanical, all those sorts of degradations over time. But I imagine that they could be quite similar given the temperature and perhaps like a salinity. But yeah, again, it was mostly the mud that saved this wreck.
Thanks for those questions. Let's give Ilya another great round of applause. So much of what our NGEN members do is new and interesting, and part of that is finding new ways to do work. So on paper, our next speaker, Paul Clerkin, is a shark scientist, and he certainly is. He's done amazing work with all sorts of species of shark that you have never heard of, but he also does incredible work with people, building relationships and friendships with commercial fishermen, which he'll talk about a little bit. A group of folks that's often sort of wrapped in an endless cycle of distrust and disagreement with scientists. So bridging that divide has been incredible work. He's a PhD candidate at William & Mary and has consulted with a variety of NGOs and regional fisheries organizations, all while making documentaries about sharks in the ocean. So I'm very excited to introduce Paul Clerkin.
Hi, everybody. My name is Paul Clerkin. I'm a shark researcher and PhD student at the Virginia Institute of Marine Science. I also operate my own nonprofit, Deep Blue Research Foundation. I've been lucky enough to work with some high-profile groups like the United Nations, some international fisheries management organizations, and sometimes I'll do documentaries with Discovery or Travel Channel. The way I've been able to be successful with some of my projects has been combining high-tech gear with age-old fisher knowledge, which is sometimes generational. So two of the projects that I want to talk about real quickly is one project I've been doing in Taiwan with megamouth sharks, and then another one in the Indian Ocean discovering new species of sharks, fish, and invertebrates. All right, so this is a megamouth shark. You can see they kind of look like a puppet. They're not really what you think of when you think of sharks, but they're real. They're very rare. They're so rare that they weren't discovered until 1967, when the first specimen was accidentally encountered. It was ensnared in an at-sea anchor. Since that time, very few specimens have been encountered, and we know almost nothing about them. They've been kind of considered too rare to study and so rare that we can't really protect them. But I knew some people in Taiwan who were reporting that megamouth sharks were mysteriously showing up in some fish markets. It would show up in one market. Most of the people didn't know what they were, their fishers didn't know, and people would just, they would eat them and not report them. They weren't going to museums, they weren't going to researchers. So we found the sea captain, and he guaranteed us that he could get us a megamouth shark. At the same time, one of my friends working at a technology company had developed an ultra deep sea tag that could collect data from geomagnetic changes in the bottom of the ocean. And then we could use those tags to track these deep sea sharks. So we were very excited. We had our team ready. We had our tags ready. The problem was we couldn't get funding because after every time we applied for a grant, it was rejected because they thought the project was impossible. After some frustration, we were able to get funding from a private donor who believed in the project, and we flew out to Taiwan. We boarded a small, brightly colored boat, and on the second night, we got a megamouth shark. So I jumped in the water, the bubbles cleared my mask, and I was face to face with this deep sea shark, which we've almost never seen alive. Yeah, this was a very cool experience. We were able to put on our little supercomputer tag. This is the first successful tagging of a megamouth shark with a satellite tag. We went out the next night and we tagged 2 more. So we were able to use that data to figure out where the sharks go, how they use their environment, and as a direct result of our work, shortly after our project, megamouth sharks were completely banned from being landed in Taiwan. So they went from being pretty much unknown to completely protected, at least off of Taiwan, which is, I think, a satisfying result of our research. Oh. Thank you. Thank you very much. But working with these fishers and other commercial fishers, we've been able to tag lesser studied sharks. It's a lot more affordable, and the fishers are able to find these sharks much more readily than if I try to go out in my own boat. The other project I want to talk about is the work I've done in the Indian Ocean. This is on a commercial deep-sea trawler. The Indian Ocean is much less explored than the Pacific or Atlantic Ocean, and there's fewer research vessels, and there's less information on where you can fish and where you can find these animals. I have a relationship with this deep-sea trawler. It's 50 years old. It's a 250-foot boat, and it's been fishing in the Indian Ocean for 30 years. This is the crew that I go out with, my friends, and some of these guys go to sea 9 months out of the year, every year, for all 30 of those years that they've been fishing out there. So I feel like that's more than half their life. It might technically make them marine mammals. Although they don't have any obligation, they let me go out with them anytime I want. I've done 3 trips, and they've been up to 3 months at sea. And we fish in this remote area, kind of south of Madagascar, about 1,000 miles from land. It takes us a week to get there, and I think when we're on the fishing grounds, the closest people to us are the astronauts when they orbit overhead in the International Space Station. Every time we go out, we discover new species. So we get rare species like this goblin shark. We've gotten multiple goblin sharks, and they've gotten them so healthy that they're thrashing, and it's hard to put them over, and I think they probably survive the descent back to the deep sea. We've gotten these weird false cat sharks, and then we've gotten a lot of new species. So we've gotten about 16 new species, and that is 1 out of every 3 sharks that we encounter are new species that have never been documented. They haven't been named. We didn't know they existed. The same is true with the bony fish. We have a lot of bony fish. A lot of these are weird. We have this barrel eye. This species has a clear head, so it can see through its own head. Mother Nature was creative with that one. But we have a lot of these rare species that are either very important for science or completely new to science. This fish is one of my favorites. I call that one a potato fish. It's just like a little potato with a tail on it. And I've shown it to a lot of different researchers at different museums, and nobody knows what it is. It has to be a new species. And the same goes for the invertebrates. We've collected a lot of invertebrates. I've shown them to invertebrate people, and they think they say they're all new. We haven't gotten along to describing them yet, but they're on the list. So, in addition to describing and naming these species, which is the first step in protecting them or at least collecting data on them, we're also taking genetic barcodes. So we'll have genetics for all these species, and that will help us study them further and also do eDNA surveys. So eDNA, which is environmental DNA, is just genetic material sloughed off by animals into the water, and you can collect that water, filter it down, and look at the genetics on that filter. And we've been very successful in surveying animals based on these water samples. eDNA is normally a very expensive process. You need expensive gear, it's time consuming, but we've recently developed this little passive sampler that you just pull through the water, and we can 3D print those for less than $5. We also made this deep sea camera rig, which is essentially a GoPro in a can with a light on a chopping board with some rope, and we attach that to the fishers' nets. So deepwater cameras are very effective for surveying. You get high-resolution images of the bottom of the ocean, and you can also see some of the species that you might not catch in nets, things that are small or highly mobile. They're expensive, though. I probably couldn't afford the cable or even the fuel it would take to lower these cameras to the bottom, but by strapping them to the fishers' nets, we were able to get this pretty fun footage of the bottom of the ocean in the Indian Ocean. So because of the remoteness of this area, I think this is probably the first video ever taken of this part of our world. We are also working with fishery museums, the fishers and technology groups like Creality and Riva Point to do 3D scans of these specimens. These specimens are rare, they're in museums, and they can be hard to access for researchers. But if you scan them, you have a 3D model, and you can even 3D print that out to have a fish in hand, and that will greatly increase the accessibility to not only researchers, but also educators and even enthusiasts, people that just like sharks and fish. You can 3D print them. So yep, we have a lot of other projects and a lot of other things going on. I can't talk about them all now. I would love to show you pictures of fish all day long, but please ask questions, come up afterwards, or contact me. My email is pauljclerkin@gmail.com, or go see our websites. Thank you very much.
Thanks so much, Paul. I think we have time for one quick question, and I know you all have lots of questions, so please do find Paul afterwards, but one quick question?
Hi, thank you so much for your talk. I was wondering if you have any concerns with how the deep sea creatures are getting caught for when we're working at the commercial trawler. Yeah.
Yes, I mean, I think it's always a concern. Their fishing doesn't seem to have a huge volume of bycatch. It's cleaner than some of the other fisheries I've worked on. That ideally it would be zero, and I think the fishers would agree. But when they catch them, at least we can use them for science. Yeah, there's a big variety and new species which I think are important to have. But yeah, obviously bycatch is bad.
Thanks for the question, and let's all thank Paul once again. All right, we've got 2 more speakers for you, and if you have ever spent time around our next speaker, you know there is no greater advocate for seed biodiversity and preservation. So as an ethnobotanist and an educator, Sefra Alexandra, our next speaker, has conducted expeditions both to preserve native seeds and restore ecological corridors with native plants. So please welcome Sefra Alexandra up to talk about her work.
What a pleasure it is to be presenting with all of you amazing folks. Good afternoon, fellow naturalists. I am Sefra Alexandra, the seed huntress on the hunt to preserve the biodiversity. Is that making a weird noise? Can you guys hear it? Okay. On the hunt to preserve the biodiversity of our earth through seed conservation. Perhaps you've heard of the Lorax that speaks for the trees. Well, to grow them takes lots of seeds, which everyone needs. As an ethnobotanist, I have conducted field work around the world, working for the global seed banks, building community seed banks on island nations after natural disasters, and what we will talk about today, replanting the living seed banks, the soils. All of my endeavors revolve around these magnificent jewels of global biodiversity, these living, breathing embryos that represent an unbroken chain of stewardship and seed dispersal by the winds, waters, ants, birds, and us, the humans. Let's take a moment to relish that seeds have germinated after 2,000 years of sleep, or that a tiny acorn can grow a mighty oak. Pretty cool. As we are surrounded by a room of scientists, explorers, and nature enthusiasts, I am pretty sure you all have marveled at the wonders of flora you meet with each intrepid step, or the meals that you prep on your journeys. Each crop that nourishes us came from somewhere. They too have ancestors, their crop wild relatives. And where the crops first emerged on the planet is where they have the greatest diversity. Potatoes in Peru, pulses and grains in India, maize and wheat in southern Mexico. This map paints an understanding of where the international seed banks lie to preserve this crop diversity. We begin to view our world and our plates as a mosaic of varied paradises found on the ground and in the canopy, medleys of agrarian triumph from cultures all around. And as you may have heard, we are losing quite a bit of this biodiversity every day. So important to preserve it in the world's seed banks. So now, as we know, natural and man-made disasters can serve quite a blow. And please allow me to be a bit lyrical as we go, as I welcome you all to the UN Decade of Ecosystem Restoration. Quite a show. The trouble is, the local plants that feed and sustain are wiped out in these instances, causing severe civil and ecological strain, such as in times of drought. The problem is all over the world, there is hardly any supply of bioregionally adapted local seed to replant fields when we have the greatest need. Now, this is not a talk of doom and gloom, for it is perhaps too late to be pessimistic, but rather, my hope in explaining the model I'm about to lay out is that wherever you do your work, there is a way to replicate this, no doubt. So here in the US, heeding the international siren call, we had to create a framework to safeguard our seeds, wild, cultivated, and all. From the EPA, the Environmental Protection Agency, this eco-regional map was formed. Seeing the boundaries from an ecological versus state line perspective was normed. Hydrology, geology, ecology became our lens. Through bug eyes, we see the connecting riparian zones from broadleaf to fens. The map guides us from a genetic resources perspective in regards to truly local native plants called ecotypes. Knowing that when we collect seeds and replant seeds within these zones, here in New York, we're Ecoregion 59, We are putting the right plants in the right place. Makes sense, yeah? So, to solve the problem of the high demand and lack of supply for ecotypic seed to ecologically restore land in severe need, we had to create a seed supply chain model. What you are looking at is how to create seed sheds to produce seed heads. We have long learned about watersheds and food sheds, so the same principles of locality apply here. We start with sustainable wild seed collection, then to the organic farmers for plant resurrection. Next, the seed hunters gather the harvest, then to seed collectives for distribution and protection. Seed hunters with permission and training go to the wild lands, monitoring and searching for sustainable size stands, making small collections of these vital natural resources to increase in farmers' fields to meet the demands. So now back to the seed production cycle. The seeds collected and protected, oh, I think we skipped one. Who did? As in art, we know how important provenance may be. So now with seeds, we track the same. Makes sense to me. Where did you come from? Who are your friends? Where is best suited to root your ends? These seeds know how to persist in these lands, and their intelligence is best suited to meet the climate's demands. And wow, our seeds' art. Here, for fun, is the bird of paradise, which hails from South Africa. Paradise seed is certainly found when you find this gem on the ground. You can kind of see it over there. So now back to the seed production cycle. The seeds collected and protected from the wild are germinated into baby plants, encompassing magnificent arcs of wild intelligence, local adaptations, and ecosystem services. 200 of each species are brought to farmers we train in the specialty crop of seed production, actually not such a pain. From cities to the countryside where there is available lands, these plants are stewarded by caretaking hands. The plants flourish and flower, a visual shower, the living seed bank thriving with life. Let's all take a nice deep breath from climate strife. Pollinators of every shape, size, and color create a buzz of ecological and symphonic splendor. The diversity of such life takes a true artist to render. These ecotypes are pollinator gold, heirlooms with stories deep and old. Right place, right time, the plants must be to feed the bees that feed you and me. The flowers bear fruit and the seeds they do come forth. This is when we seed hunters find our true north. Collecting and collecting, the bounty is rich. The ROI of seed, so much you don't have to be a snitch. Seeds we collect from the fields where they were sown. Seeds we collect from provenance known. The heirlooms of the pollinators are back, no need to fear. We have seeds coming out of every ear. Winnow and rub, clean them we do, sort of a MacGyver mission, it's true. The detective work of what each species looks like is endless fun, seeing the gems made from the sun. The seeds are back, stored and protected, for future use if lands are destroyed. For the brilliance in these jars, in my opinion, exceeds ours. So how do we return them to your hands to plant in the lands? Start a Northeast Seed Collective for farmers to sell local seed to restore local lands with their crop. These organic, eco-regional, ecotypes worth their weight in gold, full stop. A free local seed library is a wonderful way to pass these around. You return a handful of them like you would a book once the next year's seeds sprout from the ground. But wait, there's more. We want to get all involved. The botanical quest thus evolved. Now, I'm not a botanist, but a botanist. Now, dear colleagues of the Explorers Club, I would like to share with you that my expeditions of late have not been in far-off lands. It's true. Conducting this seed shed work here in the Northeast, I have seen the degradation of the landscapes here, to say the least. So backyard expeditions I do lead with these vital local seeds and plants in my steed, for every citizen scientist, young and old, to attend, for we all have a role to play in the restoring the soils we together tend. Once these seeds wiggle in their radical roots, and up come the seed leaves, their cotyledon shoots, nature has myriad dispersal mechanisms: animachory by wind, myrmechory by ants, hydrochory by water, and anthropochory by us, humans. Wherever you go, may you now look at the power these mighty, tiny little seeds bestow. We are the people of the pinch, at a pinch of time in the history of seed biodiversity, where we can either sit by and watch it erode away or stand in as seed stewards. So may I conclude with save seeds, because seeds save. The privilege of being able to help the wild lands we love lies in all of our hands. Thank you very much.
That was fantastic. I don't think we have time for questions, but definitely do come find Sefra afterwards. Thank you again. All right, I have one more speaker for you. Katlin Adamczyk is an oceanographer and a marine geochemist working for the USGS, that's the US Geological Survey, in California. She has been part of dozens of expeditions at this point around the Arctic, Pacific, and Atlantic Oceans. She is an expert on how mercury cycles through the oceans, ending up in things like tuna and swordfish, and then in us. She was part of EC50 last year, I think, and has a PhD in environmental science from Wright State University. So I'm excited to hear about deep sea discoveries.
Thank you. Hi, everyone. Today I wanted to talk about an expedition from 2023 in the Western Galápagos Spreading Center. This expedition took place on Schmidt Ocean Institute's new Falkor 2 research vessel. And the expedition was focused on deep sea hydrothermal vents. Hydrothermal vents are found in regions where there's a subsurface source of heat. So the rocks underneath the surface of the seafloor are hot. In this example on the left, we have a seafloor spreading center, so that heat is actually pushing tectonic plates away from each other. And there are cracks in the seafloor where cold seawater can seep in, and it interacts with that really hot rock. And when you heat seawater, it can change the chemistry drastically. So you then create this superheated vent fluid that is enriched in metals. And that fluid can then escape from the seafloor. Sometimes it escapes as a focused flow, which is what you're seeing on the top video. So this is a focused flow black smoker vent. The temperature of this vent was a little bit over 300 degrees Celsius. And the rock chimney structure that you see is actually minerals that have precipitated from the metals in the vent fluid. And you can't actually see the seafloor in this top video because this particular vent chimney is several meters tall. Sometimes you get diffuse flow. So rather than one focused channel, you have many smaller channels where warm fluid is seeping out of the seafloor. And that's what you're seeing in the bottom video. So on the right, it looks a little blurry. That's actually the warm fluid coming out of the seafloor, maybe 20 degrees Celsius. In both of these types of vents, we see these really amazing deep sea ecosystems. There are microorganisms that are able to actually use the chemicals in the vents to create their organic molecules, and they fuel these really unique deep sea ecosystems. The Galápagos Spreading Center was a really exciting place to work. This is the location where deep sea vents were first discovered in 1977 in the Eastern Galápagos Spreading Center over here by the Rose Garden. The line that you're seeing on the map is the spreading center. Galapagos Islands are down here. So along the line, the seafloor is very slowly spreading apart. In 2005 and 2006, there was an expedition to the Galapagos Spreading Center where they were looking for new hydrothermal vents. So they attached a sensor to a wire, lowered that sensor down close to the bottom of the seafloor, and they were looking for chemical signatures. They were looking for those plumes that are coming out of the hydrothermal vents. So they did this where you see the red line on the map, and everything that's labeled in yellow are new vent fields that they were able to discover. So once they started seeing chemical signatures of the vents, they put down a camera that they're able to tow across the seafloor, and they were able to take images and confirm these different vent fields. But there were some locations where they were seeing vent signals, but they were not able to find the vents. In 2023, we were able to go back for the first time and actually collect physical samples from these newly discovered vents in the Western Galapagos Spreading Center, and we spent some time hunting for those vents that they weren't able to find on the last expedition. So for my work, I'm really interested in mercury, and there's mercury coming out in this vent fluid, and I want to know where it goes. So during this expedition, I was collecting vent fluid with these gas-tight titanium samplers, as well as hydrothermal sediment around the vent and within the vent fields, and chimney samples, so the rock that fluid is coming out of. And so I'm looking at the concentration and the mineralogy of mercury in these different areas of the vent field to try and understand where all the mercury is going. But as I mentioned, we also dedicated some time to looking for new vents in the western part of the spreading center. And we basically picked up where the other researchers left off 17 years ago. So we started doing something called a toyo. We have sensors attached to a wire, and we move them up and down in the deep water column, kind of like a yo-yo. But at the same time, the vessel is moving. So we're towing and yoing at the same time, looking for that chemical signature that tells us there's a hydrothermal plume. It's not the most exciting work to do. We did this for almost 4 days, and I spent a lot of time sitting in front of these computer screens looking for really tiny increases in temperature. Sometimes you see an increase in turbidity that tells you there's a lot of particles in the water. That's a good sign. We have ORP sensors that tell us a little bit about the chemistry of the fluids. So after staring at this screen for over 3 days, we started to see evidence of hydrothermal plumes. We did some additional mapping in this area as well, and then using those two pieces of information, we put together a dive plan. So this is ROV Sebastian getting ready for our dive. This particular dive lasted 42 hours, and we were working at depths around 2,500 meters. During the ROV dives, I serve as watch lead from 12 a.m. to 4 a.m. I get the night shift. And so I get to sit next to the ROV pilots and guide the exploration, guide where we're going, when we want to stop and collect samples. Schmidt Ocean Institute also has a live feed on YouTube, so I'm also trying to commentate and explain what we're seeing. For the first 20 hours of this dive, this is what the seafloor looked like. So my whole 4-hour shift, I was looking at piles of volcanic rock, fresh basalts, rock walls, covered in sediment, which means they're a little bit older. We want really young rock to find these vents. So 20 hours of looking at rock and looking at mud, and then eventually we came across a couple of these. Was anyone in this morning's lecture series for the award winners? Yeah, do you know what this is? Remember the squat lobsters they showed? So we came across a couple squat lobsters, and these are animals that are associated with deep sea hydrothermal vents. And our new strategy became to follow the squat lobsters. So we started following these little creatures around, and eventually they led us to more squat lobsters, getting closer, and we've kept following those squat lobsters up this pile of rubble, and the first thing we saw were riftia, giant tube worms. And those are feeding off of diffuse flow coming out of the seafloor. We then went on to discover 5 new vent chimneys. So we discovered 5 vent chimneys and 3 diffuse sites within this region. And to be in that room and to see these animals and to see those chimneys for the very first time was a truly extraordinary experience. The squat lobsters are getting their moment of fame. This vent site is named Sendero del Cangrejo, which translates to Trail of Crabs. They look like lobsters, but they're more related to hermit crabs. And the media really ran away with this story, so a lot of the headlines that came out after this discovery were Trail of Squat Lobsters lead scientist to new vents. So we certainly appreciate the squat lobsters, but I want to highlight the human effort that went into this as well. We had an idea that these vents were there in 2005. It took 17 years for us to be able to go back and actually look again and to take samples. We think that there are about 664 vent fields across the globe. Less than half of those have actually been confirmed, meaning going down and at least taking a picture to confirm that something's there. So there are a lot more discoveries like this to be made. If you're interested in seeing more, all of these dives can be found on YouTube. Schmidt Ocean has a nice website for this expedition, so search for Western Galapagos Spreading Center. And a little teaser, you might see some of these vents in Blue Planet 3, which is coming out in 2026. Thank you.
Thank you so much for that. We've got some time for a question or two. Okay, start over here.
Thanks for an amazing talk. I was wondering, in the area of the hotspots that you showed in the Galapagos Islands, that part of the archipelago is known to be super productive, and I feel like it's mostly thought to be productive because of just like upwelling, the Humboldt Current. Are we missing this huge component to like the nutrient influx? Do you think, like, how much of it is due to these vents?
These vents are pretty deep. So 2,500 meters was the Western Spreading Center. I know as you get closer to the Transform Fault in the middle, you're closer to 1,000 meters. So I would guess no, but I don't know if anyone has studied that. I've also had some interesting conversations with the Charles Darwin Institute in the Galapagos Islands. You can also see these vents in seamounts and on the bases of volcanoes that are submerged. And I think those areas of the Galapagos are also underexplored. So I don't know about the spreading center, but maybe something closer to the actual islands could definitely be a source of nutrients, especially iron. Yeah.
How many spreading centers are there like this around the world?
That's a great question. I don't know the number, but if you've ever looked at a bathymetric map, or if you look at one later, you'll see lines going through the middle of the ocean, down the entire Pacific, down the entire Atlantic, through the Southern Ocean. So wherever you see those lines on the seafloor, those are spreading centers. I think if you lined up all of the mountain ranges on land, the length of the spreading centers in the ocean would be greater than that distance.
Wow, that's incredible. All right, thank you all so much. Let's give another round of applause. All right, 6 incredible speakers. I hope you all enjoyed that as much as I did. I am so excited to see what's next for all of these folks, what they're going to get up to. Our next generation of explorers is really an incredible group of people. I mean, everyone in the club certainly is, but these young folks bring so much creativity, boldness, innovation, and compassion to exploration, and I'm so thrilled to be able to spend time with them. So I hope you'll take some time to get to know these folks and the other young explorers you see around this weekend. A lot of our young explorers, especially those in grad school, have seen their fellowships and grants and jobs and internships disappear in the last couple of months. So plenty of our members across the world also struggle to find funding for the amazing work that they're out there trying to do. So I encourage you this weekend to talk with these folks and hear their stories. And I really hope that we can, as a club, find a way with our fellowship to keep moving exploration forward for these folks. So I would like to ask all of you to come back up here for me, and we're going to grab some photos and give a chance to say thanks to these 6 wonderful speakers. All right, so we are going to give some certificates for our speakers. So we'll start with Sefra. And Paul Clerkin. And Hailey Shchepanik. Monique Sosnowski. Zeff. And Katlin Adamczyk. That was wonderful, all of you. Thank you all for coming, and please do come chat with all of these wonderful folks. I know you have more questions in you. So thank you again, and enjoy the rest of the weekend.